Hello and welcome back to this NPTEL course titled Quantitative Methods in Chemistry. We will be proceeding on week 5 of the course now, and this will be the lecture 1. Before we proceed on this week's lecture, let us quickly recap what we learned in the course in the last week. So last week we got introduced to different statistical tests such as z-test and the t-test. Similarly, we got introduced to the concept of hypothesis testing, and we also understood what are the potential sources of errors that occur in hypothesis testing. And finally, we also learned about applying q-test to identify outliers in our data. So with this background, we are all set to embark on next level of applying these understandings to what is known as analysis of variance. So this week we will be essentially focusing on the analysis of variance, and we will get introduced to the need for conducting analysis of variance, which is abbreviated as ANOVA. We will get introduced to the concept of ANOVA and protocol of doing single factor ANOVA. So we will understand what does this term mean, single factor, and how do we apply analysis of variance to obtain least significant difference when comparing means of multiple samples. And then we will conclude this week's lecture by taking a few examples of applying analysis of variance and least significant difference on different test samples. So let me begin with the discussion on what is the need for conducting analysis of variance. So far in the last week we saw that we can apply t-tests to compare differences between two samples or populations. So the obvious question is, can we extend the t-test to compare n different samples? Suppose we have 5 or 10 different samples that are being compared, and within these samples we have individual readings. Can we apply t-test to compare these samples and arrive at a particular inference? So the answer to that question is no, we cannot apply t-test on very large number of samples, because we got introduced to the term of error in hypothesis testing. That is, when each pair of data set is being compared, we are committing either type 1 or type 2 error based on the confidence level we are at. So for example, we learnt in the last week that at 95% confidence level, we still have 5% chance of committing type 1 error. And type 1 error is when we are rejecting H naught or the null hypothesis when it is actually true. So the important point or take home message is that as the number of data sets increase, the chances of committing type 1 error also increases. So we will not be able to apply t-test on a large number of samples. And for example, I have calculated what is this error if we are dealing with 10 samples at 95% confidence level, and we are applying t-test and comparing different pairs of samples. So the overall probability in this case of committing type 1 error will be 1 minus 0.95 is our confidence level to the power of 10, which is the number of samples that are being compared. So when we do these calculations, we figure out that there is about 40% chance of committing type 1 error. Now this is quite significant, and that means that any prediction that we make or any inference that we have based on this statistical analysis will be erroneous by about 40%. This cannot be ignored, and that is the reason why we need to apply ANOVA in such cases, because applying ANOVA reduces this error by condensing the comparison to two numbers, and we will see as we proceed through the lecture how this is done and what results do we obtain through analysis of variance. So let us get introduced to analysis of variance a little bit. So there are a certain presumptions, actually three main presumptions that are being made when we apply analysis of variance on a particular data set. First of these presumptions is the concept of normality of data sets. So what we imply here is that all the populations that are being compared are presumed to follow Gaussian distribution. So we got introduced to this concept of Gaussian distribution of large data sets early in this course. Another point here is that of independence of errors. Now what does this imply? This implies that the samples that we are comparing are randomly selected and are indeed independent of one another. So the errors that propagate in sample 1 will have no bearing on the errors that are present in sample 2 or sample 3 and so on. Another very important concept which is presumed here is that of equivariance. So what is presumed before we perform analysis of variance is that the variances, which is the standard deviation squared, please note the square. So the variances of the populations being compared are presumed to be similar. In other words, the largest standard deviation of a particular sample should not be greater than 2 times the smallest standard deviation of a different sample. So this is one important exercise that we need to do before we apply analysis of variance on our samples. So in nutshell, in analysis of variance, what we attempt to do is to compare the fluctuations in variance between the samples with those within the samples. So comparison between with within. So we will see how this is done and what does this yield us to. At this point, it is important that we look into how variances are compared in statistics, and for that we employ a test which is called the F test, or the F statistics is employed when we are comparing the variances, or in other words the precision of two measurements. So just a brief aside, the F statistics is named after Ronald A. Fisher, who made very significant and seminal contributions in the field of statistics. So to honor him, the F test is named after him. So here the protocol that we apply for undertaking the F test is that we establish a null hypothesis as always. So the null hypothesis presumes that the population variances being compared are equal. In other words, if we are comparing two samples S1 and S2 and they have a standard deviation small s1 and small s2, then the variances, that means the square of the standard deviation values, are similar. Next, we apply the test statistics. So we calculate F test value, which is nothing but the ratio of variance 1 by variance 2. Now again, please be aware and please note that we are squaring the standard deviation to compare the variances, which is what variance is actually about. So also note that when you are calculating the F test value, you keep the larger standard deviation value on the numerator. So this is a protocol that needs to be followed to obtain F test value, and this F test value will be compared again with a statistical table of F, and we compare it with a critical value of F, which is now found at a particular confidence level. And now there would be 2 degrees of freedom associated here, one with regards to the sample 1, which will be at the numerator, which is often denoted by mu1, and similarly there will be a degree of freedom that is associated with sample 2, which is denoted as nu2. So just to give you a quick view of what these statistical tables look like, this is one F distribution table which is shown here, and the critical value of F are tabulated at 5% significance level, or in other words 95% confidence level. Now this is the confidence level which we have been working quite often in this course. So I also want you to note that you have nu1 value, which is in this case tabulated up to 20 degrees of freedom, and similarly you have a nu2 value which is tabulated up to 1000 degrees of freedom. So this is the table that we have at 95% confidence level. Now going to 99% confidence level or 1% significance level, we have another table which is again similarly tabulated with the 2 degrees of freedom being mentioned in the table. So we can utilize the F table and conduct the F test to compare the variances or the precisions between 2 samples that are being compared. Let us actually apply this F test to compare the precision of 2 samples. So suppose we are dealing with a standard method which was based on say 1001 data points for estimation of say sodium ions in water, and this standard method has a standard deviation value of 0.8 ppm. Now we come up with a new method which is based on only 21 data points and has a standard deviation value of 0.21 ppm. So we see that for the reference or the standard method, the standard deviation value of 0.18 seems a little lower than the standard deviation value of the new method, which is 0.21 ppm. So the question that we need to answer is, can we say at 95% confidence level that the new method is more precise than the standard method? So how do we approach this problem is that we first apply the null hypothesis, and the null hypothesis presumes that the standard deviation for the reference and the test methods are similar. That means the 2 methods are indeed similarly precise in giving their results with regards to the sodium ions present in the samples being analyzed. Now we calculate the F test value here. So again I want you to note that we have put the larger standard deviation value of 0.21 on the numerator, and that is squared. Similarly, the smaller standard deviation value of the reference method of 0.18 is put in the denominator, and it is again squared. When we undertake this calculation, what we get is a F test value of 1.36. Now this F test value needs to be compared with a F critical value of 95% confidence level and 20 degrees of freedom for our new method, which is giving a higher standard deviation value, and 1000 degrees of freedom for the old or the reference method, which was giving a lower standard deviation value of 0.18 ppm. Now let us quickly go back to the F table at 95% confidence level and see what the values would be. So at 95% confidence level, nu1 of 20 and nu2 of 1000 gives us a F critical value of 1.58. When we insert this value here and compare the F critical value with the F test value, what we obtain is the test statistic is less than the critical statistics. So what we can say is that under these conditions, the null hypothesis cannot be rejected. That means at 95% confidence level, the two methods being compared have similar precision. So we see how the F test is being applied in comparing the two different methods or samples. Now let me take you to the concept of single factor ANOVA. So we will get introduced to the single factor ANOVA in this lecture, and in the next lecture we will be applying the single factor ANOVA on a real data set. So the questions that we can address with single factor ANOVA can be that whether the yield of a reaction changes with temperature, or if a spectral profile of a molecule changes with pH. So the terms highlighted in red here are our variable, and this is the also known as the factor in analysis of variance. So the factor is being varied in our analysis, and what we do is we have the spectral profile or the yield of the reaction as the different levels. That means at a particular temperature we calculate the yield at multiple times. Similarly, we change the temperature by a certain unit and again calculate the yields multiple times. So the yields of the reaction in this case is the level that is being changed. Now the response is nothing but the change in the value of measurement being performed. For example, when we are comparing the spectral profile of a molecule with change of pH, it is the wavelength which will be the response, and the spectral profiles generated at different pH will be the different levels, and pH is a variable here, so it will be the factor. So a single factor ANOVA in fact implies that there is only one factor that is being varied in these analyses. So, what we will do is we will undertake a detailed example of how analysis of variance is to be conducted for a single factor in the upcoming lecture.